Hello, a warm good morning to all of you. I feel honored to welcome all myself, Dr. Binu Ramachandran, Assistant Professor, Department of Zoology, and Principal Investigator, Smriti Brain Research Lab, Christ College. And this is indeed a fantastic opportunity, such a wonderful virtual gathering amid the pandemic scenario. I'm happy beyond measures to welcome you all to Synapse 2020, organized by Smriti Brain Research Lab, Department of Zoology, Christ College, Irinjalakuda. Synapses 2020 become each one of you to a phenomenal session of scientific enlightenment, furnishing you with fresh insights and novel concepts. Let me invite Dr. Sia Joshi, Vice Principal and Head of the Department of Zoology, for welcome address.
Dean of Science, Dr. Basic Corby, and the IQAC Coordinator, Dr. Robinson from Mississippi. Here's me inviting the resource person of the day, Dr. Vinita Bharat, who is the part of Neurobiology Division of Stanford University, the United States of America. I am inviting Dr. Gloria to CMI for his blessings for the international webinar on crash disposal of degenerative diseases. Thank you.
Hello, now I request Reverend Father Jolly Andrews, Principal, Christ College, to deliver the presidential address.
A warm good morning to everyone. Am I audible?
Yes, yes.
Thank you. So, the chief speaker of the day, Dr. Vinita Bharat from the Stanford University, United States. The convener, Dr. C.O. Joshi, the Vice Principal and the HOD of Zoology Department. Organizing Secretary, Dr. Binu Ramachandran, the Department of Zoology. Respective professors, teachers, colleagues, and those who have joined for this webinar, Synapses 2020. I'm here to speak a few words about our college. The Christ College was started in 1956 by the Devamata province Lives of Mary Immaculate and an indigenous religious congregation. It was founded way back in 1831 by St. Kuriakos Elias Chavra. He was a very saintly priest, a versatile genius who always saw education as a tool of liberation and development. The Christ College is affiliated to the University of Calicut and re-accredited by NAC with the highest grade A, and is dedicated to Christ and has its motto, Jivita Prabha, which means the light of life. Perhaps the very motto of this college has got a special meaning in this time. The light of life, where we are in a way struggling hard in order to promote life in this special scenario of COVID-19. Now, the Christ College became one of the prominent epicenters of the academic training in the country, thanks to the visionary leadership given by the first principal, Padma Bhushan Father Gabriel, he himself being coming from the Department of Zoology. The Christ College is at present 26, 27 UG programs, 16 PG programs, and 11 departments having PhD research sites, and now being the Zoology, the leading the department which is offering the PhD. The college also offers 21 certification programs which are of immense use in the job market placement, and has at present 3,860 students, the largest number in Kerala. The college has also entered into MOUs with the University of Sudan, South Africa, Leibniz Institute for Applied Geophysics, Germany, which presents the students with an international exposure with an eye for higher research. Now, the Christ campus is a lush, green, beautiful one, and a casual walk through the campus will definitely help one to strengthen the immune system. We think of health in a very special way in these days in the wake of the present pandemic crisis. Now, I'm very happy to offer my felicitation as well as to speak a few words about the today's International Webinar-Synapses 2020 that has been organized by the Department of Zoology, Smriti Brain Research Lab. The topic of the today's discussion is the problem of trash disposal in neurodegenerative diseases. And Dr. Vinita Bharat from the Stanford University, United States, will be always addressing us. I especially congratulate the Department of Zoology, the Vice Principal and the HOD, the convener of today's program, Dr. C.O. Joshi, the organizing secretary, Dr. Binu Ramachandran, who have taken all the efforts in order to make this wonderful webinar a possibility. Especially in the wake of the COVID-19 pandemic, we need to, in a way, creatively as well as positively think about the topic which you are discussing, how we have to dispose the trash so that we can, in a way, prevent the occurring of the neurodegenerative diseases. I hope we will have a wonderful time. May God bless you all. Have a nice time. Thank you.
Thank you, Father. Dear participants, I would like to request all of you to mute your audio and video during this session. And now, it's my pleasure to introduce our resource person, Dr. Vinita Bharat. Here is a short bio brief of Dr. Vinita to best of my knowledge. Dr. Vinita completed her bachelor's in biochemistry from University of Delhi. Then she was a graduate fellow at the International Max Planck Research School in Neuroscience, Göttingen, Germany. And later, she was a postdoctoral fellow at the European Neuroscience Institute, Göttingen, Germany. Currently, she is a researcher and a scientific illustrator at the Department of Neurosurgery, Stanford University, United States. Vinita is not only a brilliant scientist, but also a creative genius. Her Fuzzy Synapse, an interesting and engaging science cartoon platform, helps to communicate science in a fun way to the public. Ladies and gentlemen, please join me in welcoming Dr. Vinita Bharat to Synapse 2020.
Thank you so much, Binu, for such a nice introduction. Good morning, everyone. It's so nice to see so many of you here in the morning. Firstly, thanks. Am I audible?
Yes. Yes,
okay, perfect. Firstly, thanks to the Christ College for hosting such a beautiful international webinar, Synapses 2020. And a special thanks to Dr. Binu Ramachandran, with whom I have shared my PhD days in Germany. So a very, very special thank you to for inviting me to today's talk. Okay, without further ado, I would like to start my talk. So I will be sharing my window. Just give me a second. Perfect. So I hope you all are able to see my window and presentation.
Not yet, Vinita. We're just waiting for that.
Is it visible?
No, not yet.
No.
Oh, just a second. Is it visible now?
Yeah, it's loading now.
Yes, it is.
Oh, okay, perfect. Perfect. Thank you. So, yeah, so the topic of my talk today is the problem of trash disposal in neurodegenerative diseases. I'm a postdoctoral researcher in Professor Shinan Wang's lab and Department of Neurosurgery at Stanford University. And in today's talk, basically, I've divided today's talk into 4 different segments. The first would be talking about neurobiology in general, giving a little bit introduction about how what is brain, what are neurons. Then we will be moving forward to mitochondrial dynamics in neurodegeneration, which is the focus of our lab. Then I would be talking about a mitochondrial protein called Mito, which is involved in neurodegeneration. And lastly, I would be talking about some potential therapeutics to Parkinson's disease. I would request everyone to, if you have any question throughout the presentation, please feel free to write in the chat window so that I can take it later. Okay, so this is the black screen. And if everybody can see, there is a small white dot here. This dot is actually a fly brain. And if we move from fly to mouse, it looks like this. And further to human, it looks like this. So you can see that if we compare from fly to human, the brain size increases. And as the brain size increases, the complexity of the brain also increases. But the most common structure, which remains throughout different species, which is same, is neurons. So when we talk about neurons, neurons are the fundamental unit of the brain. They are basically, they are having this peculiar structure of having a cell soma from where the proteins are synthesized. It acts like a factory in which various kind of proteins, molecules which are needed for the neurons are synthesized in cell soma. Then there are dendrites and axons through which these materials which are synthesized in cell soma are released. And this release trafficking in the axon is basically in 2 directions, anterograde directions or retrograde direction, which means that molecules which are coming from cell soma to the terminal are anterograde and vice versa is retrograde. And then there is axon terminal from where the proteins are released. So this is a very basic neuronal biology where it shows that how a basic unit of brain works. And you can imagine that we just don't have one of this. We have like millions of neurons talking to each other at similar timings. So, and it's so precise that the function that we as a human work. So when we draw a neuron, we just draw a single straight axon. But in reality, neurons have long axons, as you can see on the right side of my window. So neurons have such a long axon, so you can imagine that the things which are synthesized in the cell soma has to travel all this distance to reach to the terminal. And while they're moving, they have to be spatially, temporally regulated so that whenever there's a need of something, it is present at that time to suffice the functionality of the neuron. But how does this everything works? It works because there is a need of energy. Neurons can't function if there is no energy. So for proper functioning of neurons, with having so many in our brain, we need energy and neurons need energy. For neurons, the energy comes from an organelle called mitochondria, which is the focus in our lab. So mitochondria are basically powerhouse of neurons. They are there, which act as an energy packet for cell. They produce ATP. They are synthesized in the cell soma and are then transported into dendrites and axons. They maintain calcium homeostasis and also generates reactive oxygen species and signals apoptosis. So you can imagine that mitochondria plays such a crucial role that not just the energy delivery, they also maintain calcium buffering, they also maintain the apoptosis, signals apoptosis. So there are so many functions entitled for the single organelle in the neuron. Hence, it's really, really important that a mitochondria is healthy inside a neuron to make the neuron healthy. So if there's any defect in the energy source, it can lead to neurodegenerative diseases, which means that neurodegenerative diseases is nothing but the progressive loss of structure or a function of neuron, leading to death of neurons, which happens in many diseases like Alzheimer's, Parkinson's, Huntington's, etc. So for proper functioning of neurons, you need healthy energy. And if there's a defect in that energy, it leads to neurodegenerative diseases. So now the question comes that why is mitochondria playing such an important role? So you can imagine that in a healthy state, there is healthy mitochondria. And as we had discussed before, that neurons have long axons. So in that long axons, you need like multiple mitochondria. So mitochondria should be healthy. It should transport very nicely. It should be distributed wherever it's needed. And when there is any damage to these mitochondria, it should be removed quickly so that it doesn't waste energy of a neuron, right? So healthy mitochondrial transport, distribution, and damaged mitochondrial removal, these two plays important role to have a functional mitochondria inside a neuron, which leads to healthy neuron and which leads to healthy state or healthy condition. But on the other hand, if it's a diseased condition, then there's a stalled unhealthy mitochondria, which means that the mitochondria are actually unhealthy, but they're still there. And the mechanism by which these damaged mitochondria are removed is actually having some problem, because of which there's a dysfunctional mitochondria leading to loss of healthy neurons and a disease state. So this is the basic important, like this is so important to understand that the mitochondrial dynamics is actually handling the healthy or a non-healthy state of a neuron. So our lab is basically focusing on studying these mechanisms. So here's a time-lapse video, which means that I have imaged these neurons under a microscope. These are actually human neurons, which are induced from stem cells. And there are two panels you can see on left or right. In left, I have labeled these neuron, which has a cell soma, a dendrites, and an axon. I have labeled this neuron with a GFP, which is basically labeling it green. And on the right-hand side, I have basically labeled mitochondria with a red color label. So that's why all these red pigments which you're seeing on the right-hand side is actually mitochondria. So this is a healthy patient cell, like healthy neuron. So if I run this video, you would see on the right-hand side that there is trafficking of mitochondria. These small dots which are moving is actually mitochondria, actually like moving in time lapse in a neuron. Whereas if I compare a diseased neuron, which is here, which is done in the same fashion, this was a patient neuron, in which we have on the left-hand side a GFP marker, on the right-hand side mitochondria labeled with M-cherry. And in this process, in an axon, you can see that there are very few, like most of them are stalled actually. Nobody is moving, no mitochondria is moving in that axon. So here in these two time-lapse videos, you can easily see that in one healthy condition, mitochondria is freely moving and in a regulated way. In the second disease condition, mitochondria are stalled, which means that the mitochondrial trafficking plays a huge role in making the condition like healthy or unhealthy. So with this, in a lab, we focus on understanding mitochondrial dynamics. And our major focus is to study the transport and the clearance, like how the mitochondria are transported in the neuron and how are actually they cleared from the neuron. So this is a very beautiful video, which I really love. So this is in general showing trafficking inside a cell. It was taken from Inner Life of Cells. So it's a huge video, but this is a small snippet. Here you can visualize that how vesicles or huge proteins are transported inside a neuron. If I play this video, you would see, like when I play, just remember that these long tube-like structures are called cytoskeleton. And this blue color thing is called vesicle. So you can see the beauty by which these vesicles move. It's so beautiful. Like there's this huge blue particle which is moving on these tube-like structures called microtubules with the help of this motor protein which walks on the microtubule. So this is how in a cell long-range transport happens, and neurons having these long axons, such transport plays a huge role. You can see like how these things are moving so precisely on the microtubules. Now, the same method is done by neurons in mitochondrial trafficking. So in mitochondrial trafficking, you have a mitochondria, a healthy one, which moves on tubes called cytoskeleton or microtubules, and through these motor proteins. So they cover long distances by moving like this. This is a small cartoon which I drew just to make the concept simpler. So here you can see, I have focused on this, I have marked this something called Miro. So Miro is actually a protein of our interest, and Miro is a protein which is present on mitochondria, and it actually helps in moving the mitochondria on cytoskeleton. So this is very trivial way to show it. So if I have to show you exactly how it looks, it is something like this on the right-hand side. So you have a mitochondria on which you have a Miro protein, which has calcium binding domains, GTPase domains, and this Miro protein is actually bound to Milton, which is further binding to kinesin motor protein. So you can see that on the left-hand side, it's just a simple way to show it. On the right-hand side is actually how proteins assemble together to move mitochondria on microtubule. Okay. So here is a small video animation which I showed just to see how mitochondria traffic inside a microtubule. So you can see that if I play this video again, that mitochondria is actually moving from one place to another, carrying this whole protein complex from Miro, Milton, and kinesin. They move together with mitochondria from one place to another. This is a condition of a healthy state. Like in a healthy neuron, this is how it works. But what happens when there's a damage or like something happens to a mitochondria that it becomes unhealthy? So we see that it's moving fine. Then there is some damage happening because of which a mitochondria becomes unhealthy. When it becomes unhealthy, what happens is that the mitochondria is stopped inside a neuron. It doesn't move around. It stops where where the damage happens, and it just stalls around in that area. Then what happens is that the 3-4 proteins come together and attach to this whole complex. And in our lab, we have shown that it's LRRK2, Parkin, and PINK1. These 3 crucial proteins come and just bind to this whole complex, causing Miro to move out of the mitochondria and go to degradation. So if I play it again, so the mitochondria was moving fine. Then a damage happens because of which the mitochondria are stopped on microtubule. Then there are these 3 proteins which comes into play. There are multiple proteins, but these are the proteins which we focused on in the lab. And you can see that these 3, PINK1, Parkin, and LRRK2 basically comes and holds this thing, leading to Miro degradation in proteasome. And once the Miro is removed, then the mitochondria is actually lysed, and the process is called mitophagy. So this is the whole cycle of how mitochondria traffics, stops, Miro is degraded, and basically mitochondria is lysed in a neuron. And this happens in a healthy condition. So in 2016, we found in a lab that mutations in these 3 proteins, which we discussed before, PINK1, Parkin, and LRRK2, actually causes familial Parkinson disease. So talking about Parkinson disease, it's one of the most common neurodegenerative disease. And on the left-hand side, you can see that a Parkinson patient basically has resting tremor, has postural instability, rigidity, has slowed movement, that a patient is generally, like a Parkinson patient would be generally bending forward, and there has a reduced arm spring. So these are all Parkinson symptoms, like which you can see in patients, like, and it's an early onset disease, and generally like age is a risk factor for Parkinson disease. So you can see that how it, like Parkinson disease looks like in a patient. And as I said, it is one of the most common neurodegenerative disease. But the striking part is that only 5% of PD cases, Parkinson disease cases, are familial, which means that only 5% PD cases have a genetic inheritance, which we know. For example, we know that it's from Parkin, PINK1, or LRRK2, but only these and other proteins like GBA, SNCA. But these proteins basically are just 5% of the whole population. Rest 95% are actually sporadic, in which we don't even know the cause of Parkinson disease. So because of such a thing, there's an unmet need of PD treatments, that there's a high rate of misdiagnosis because most of them are sporadic cases. There is no effective treatment of PD till date, and there is no reliable biomarker or target known for PD condition. So these challenges actually prevent therapies from bench to bedside, that you can't, because if you can't diagnose, you can't treat a PD patient. So in our lab, when we figured out that mutation in these 3 proteins are causing familial Parkinson disease, we thought that the common thing in these 3 cases were that there was a defect in mero degradation, because of which mitochondria was stalled, and there was a defect in mitochondrial trafficking and clearance, which means that these 2 defects was actually could be the causal for the Parkinson disease. So we asked a question: can we use mero degradation to measure the disease? Because mero degradation plays a huge role in getting the mitophagy step, like in moving forward in the clearance of mitochondria. So we asked the question: can we use this mero degradation defect to measure actually the Parkinson disease? So we did a huge screen, and we used skin fibroblasts, which is easy to take from patients, and we did a large screen where we had 22 control group of people, which means 22 healthy patients, in which 12 patients were age, gender, and race were matched with and are recruited from the same cohorts, and 10 were done with a neurological disorder including HD and AD, which is Huntington and Alzheimer's. So we combined these 22 controls, and then we had a 71 Parkinson patients from different sporadic and familial cell lines. So we had like patients which had LARC2 mutation, for example, or a PINK1 mutation or a Parkin mutation, but also we had some conditions where we didn't know what the mutations were, which was sporadic. So we had these 71 patients together, and we also took 3 patients which were at risk. So we did this huge screen to see if the mero degradation is actually a phenomena happening in PD patients. This is a heat map which we got. You can just look at this is too much data in one slide. So we will just look at the top red panel, and this is a heat map. So you can imagine that the red ones, which are bright red, are actually mero protein is present, and the black ones are the one in which mero protein is degraded. So the top 20, which is healthy, HD and AD, top 22 people have the degradation. Like you can see that on 0 time, you have red, but when the damage was happening, because of CCP treatment, it turned into black, which means that the mero was degraded from in these conditions, healthy condition. When we talk about Parkinson patient, which was 71, you see that we had a success rate of 93%, that in 93% of the cases, we saw that mero was actually still there, was not degraded in these patients. So this came as like mero could be a common denominator in all these PD patients, because irrespective of being a sporadic or a familial, mero was actually acting as a marker to figure out whether it's a PD patient or a healthy patient. Then after this, we actually collaborated with a company called Atomwise, and we basically predicted like around a library of like 6.5 million small molecules, and we narrowed it down to 11 and then to 3 and then to 1. Basically, we saw like if these small molecules can bind to mero1 and can eliminate this mero defect in PD fibroblast. So here, this was a small molecule which we used in the lab after the whole screening done by using AI, and we found out that this small compound is actually helping in reduction. So on the right-hand side, I will try to explain the data. So this is a Western blot where if there's a band, it shows black. If there is no band, then it would be no band, like either a slight band or reduction. So in the top case, in the healthy condition, the top 4 condition, you can see that when the damage was happening, for example, when CCCE was added, which is to depolarize mitochondria. So in the second case, you can see that mero was degraded, that there was no mero present. Similarly, when you added reducer, it didn't change. It was still not there. So in healthy condition, you see that mero is actually degrading. But in Parkinson patient, you can see that when reducer was not added, when this compound was not added, you can see that mero is still present in these. But when this compound was added, then mero was reduced. So, and we have control here with GAPDH. So you can see that this mero reducer actually helps in degrading mero1 in Parkinson patient. This was actually done in fly. This panel where you can see that when in flies we treated them with this same compound, you can see that in 0 condition there was a good band for mero, but with the addition of this drug, mero is decomposed. So with this panel, we saw that this compound is really affecting and eliminating the Miro defect. We also checked Miro reducer rescuing Parkinson phenotypes in human neurons, which we grow in our lab, the iPSC-based neurons. And also we checked an in vivo PD model, like fly model, that Miro-1 reducer was actually helping in rescuing the Parkinson phenotype. This was a paper we published in 2019, December 2019, showing that Miro-1 actually can mark Parkinson disease subset, and Miro-1 reducers actually rescues the neuronal loss in Parkinson model. So if anybody is interested in reading to the article or asking more about this article, I would be happy to answer later in the mails or after the talk. So to summarize so far, what we have discussed is that in healthy condition, you have a mitochondria, which has Miro protein, moving on with motors on a cytoskeleton. If there's a damage happening, then the mitochondria get off from the motor protein and basically goes into trash, like it gets sliced. But in unhealthy condition, what happens is that this Miro bind to the mitochondria is actually still stalling on the cytoskeleton, because of which there's a problem in trash disposal, which means that the mitochondria is still there in the neuron, and because of which is eating up the energy, leading to neuronal loss or loss of function. So all these conditions is happening because there's a trash disposal problem in the unhealthy condition. I think I will take 5-10 minutes also to talk about precision neurology for PD. So nowadays, like what we are focusing on, or what we believe is that because PD is such a multifactorial, multi-causal disease, that there are more than one causes for each individual patient. And also there are so many contributing factors like environmental, mutation, immune response, another disease maybe. So PD is such a complex disease that it actually invites a precision neurology treatment, which means that a customized treatment for each individual. For example, if a PD patient if we do a proper profiling of a PD patient, which is based on environmental factors, genetic factors, whether it has what kind of mutations, or if it has other diseases or something, we can do a proper profiling of a PD patient and identify what all mutation it has. For example, in this PD patient, for example, it has a LRRK2 mutation, Miro-1 defect, or the conditions are suitable for deep brain stimulation, which is one of the treatment which is given to PD patients. So if we know that these are the mutations happening in PD patient, we can actually design a cocktail therapy for such a patient rather than giving the same treatment to all the PD patients. So we can design a cocktail therapy according to the patient. For example, in this case, we would be using LRRK2 inhibitor, maybe Miro-1 reducer because there was a Miro-1 defect in this patient, deep brain stimulation, and L-DOPA. So I think in the future, rather than giving the same treatment for each patient, there's a need to give precision, they need to give customized, personalized medicines to each patient. And Miro-1 could act as a common denominator to mark which patient has to be given which things. Like you can do a quick screen, because it's easy to do. It's from skin fibroblasts. You can actually check whether a patient is even early onset can be detected by Miro-1 reducer defect. So you can actually see if a patient has the Miro-1 defect. And if it has, then a profiling can be done and the subsequent cocktail mixture for the therapy can be given to such a patient. With this, I think I would like to close my talk. This is a lab picture where in the center we have our PI, Professor Shinan Wong, and this is us in the lab mates. And this is a Twitter handle if anybody wants to follow what papers or what progress we are doing in the lab. Also, if you have any questions regarding the project which we can't cover in this time frame, feel free to ask me. Thank you, and wishing a positive energy to everyone.
Thank you, Vinita. Thank you, Vinita, for this highly informative, insightful, and inspiring presentation. And now this session is open for discussion, and we expect an active participation from all of you. And please come forward for your questions. And now, and also you can text your queries in this chat box. And now we have a few questions. And let me ask one from Dr. Nixon Abraham. Which disease condition is this? So he's asking actually, okay, one question is about the what about the effects of any toxicity of products formed by the decomposition of Miro?
Sorry, if I get the question correct, is it like if there is a if there is anything else apart from the Miro degradation, like if there is any toxicity which we see?
Yes.
Is that
Yes.
Oh, okay. Oh, great. It's a very good question, first of all. So basically, we didn't see any, we have checked other mitochondrial properties, like, for example, checking the TMRM staining, doing other mitochondrial studies, and we didn't see that everything else was functional. So Miro defect is the only, like, using this Miro reducer, it doesn't change the mitochondrial property.
I see.
If that answers it.
Yeah. Can I ask a few more questions? So you were showed a neuron under the disease conditions. Which disease condition was this? At the beginning when you were...
Oh, that was basically APP induced. So that was not a Parkinson, but a Alzheimer APP induced condition, like APP induced protein, like
the culture neurons.
Yeah, it was basically an eye neuron. So we from stem cells, it was induced into a neuron. Yeah.
If I, Binu, can I ask?
Yes, please.
Okay.
Yes.
Okay. So in Parkinson's disease, the disease condition is mostly caused by fibrils or the oligomers formed inside the cells, or also extracellular formation, right? So the molecule that you have designed, that design was based on what property of Miro?
So basically, we look whether the molecule can bind to the GTP domain, like the GTPase domain, which is present on the Miro. If it can bind to that site, and it can help in the degradation of the Miro protein. So these molecules, this whole screening, you can if you want to go deep, we can go through the paper, but this screen was done to basically see whether these small molecules can bind to the GTPase domain of Miro. And if they can bind, then basically there was a stimulation done, and out of that top 11 compounds we tested in the lab, out of which this compound 3, or which we call in a paper Miro reducer, is actually doing the correcting the defect.
So if I understood correctly, this Miro is just one protein, right?
Miro is the one protein, yes.
One protein. So when it comes to in general, when it comes to the disease condition, so what about all sorts of aggregates that can be formed inside or extracellularly?
Yeah, basically our lab showed that even in alpha-synuclein aggregation, Miro is involved. So we are basically focusing on PD models right now, Parkinson model right now, and we have seen with alpha-synuclein and with the PD different mutation, like the familial mutations, this is a common denominator.
I see. So this would work for also for the synuclein aggregates, I suppose. Or you still have to test it.
Yeah, exactly. We have to still test it. We haven't yet tested that. Yeah.
I see. As you have done lots of dynamics studies, which is the best marker for the mitochondrial dynamics in vivo?
For example, for trafficking, you can look for mito M-cherry, like any mitochondrial M-cherry works really well, which I have used and mitochondrial, like anything, GFP M-cherry, you can label with any tag, but mitoprotein really works well with the mitochondrial tag to study the mitochondrial trafficking, especially.
Yeah, so just one last question. So when you work with the cell cultures, and if you have an in vitro model, and if you are putting any sort of aggregated forms of synuclein or whatever the protein that could be, and then the problem is it acts very fast, right? And then under those conditions, for example, you can think of the neurite collapse. Under those conditions, if you want to really track the mitochondrial dynamics that is happening, how would you go about it? What would you suggest?
One thing, like I haven't done this experiment myself, but what I could suggest is that if suppose you have aggregate formation, like if you have a molecule which does that, for example, ABP protein overexpression or something which really enhances the aggregation in neurons, if you can do a dose dependent, for example, if it gives it enough time to do some mitochondrial analysis, still increasing, like for example, overexpressing a protein, and giving it time, and then in the same time frame, if you can add the CCCP drug to damage mitochondria in vitro, or for example using FCCP, there are many drugs which comes to depolarize mitochondria, for example. So in that time frame, it gives you a time frame if you use a dose dependent curve, or like if you basically overexpress a protein, so it gives you that time frame, if that helps.
Okay.
Yeah. Mhm.
Okay, thank you very much.
Okay,
Yeah.
thank you, Dr. Nisikan. And now we have another interesting question from Chandni Pradeep, and she was asking about, are there chances of Parkinson's in developing in young ones especially, or in
There
young
are
people actually?
there is an early onset of Parkinson disease. Hence, we thought that an early diagnosis, for example, this like with the recent published data which we have, that for example the Miro1 defect, if we can check it in the starting condition, when we have like if we can check it in the early onset time, then we can actually see if the defect is present. Then we can move forward and actually give them timely treatments for it. So it is possible, yes.
Okay, and I request all the participants come forward with your questions. Please feel free to ask. No? Hello? Please come forward with your questions.
Oh, there is a question which I see here with Dhilna Sani, which says that is trash disposal problem seen in all neurodegenerative diseases? I think yes, because if a neuron have crash, for example, the damaged any organelle, for example, if it has such a kind of like accumulation, and if it's not clean, then you can imagine that the neurons won't be functioning properly. So I would say yes. Sitara Nasir is asking that are there any side effects of Mito1 reducer? As I discussed before, that in our experiments we checked that mitochondrial dynamics is not changed with Mito1 reducer, so there is no such side effects which we saw in our experiments. But of course, it's further to like check it. It's further open for research.
Okay, thank you, Vinita.
Thank you, Binu.
There is one more question about the role of Miro in apoptosis by Abhilash.
As far I know, Miro is not involved in apoptosis, whereas mitochondria is. But if there is a function of Miro, I'm not aware of that. Yeah.
Okay, thank you. And I think we have no more questions left. And we are coming to a close of Synapse 2020. And I believe our resource person has answered every questions in a convincing and reasonable way. So we come to an end. It was really a fascinating and engaging time that we all shared. Now it is my pleasure to thank the entire team behind Synapses 2020 who have put in their valuable time and effort to make this webinar successful and memorable. I take this opportunity to thank Dr. Vinita Bharat from the bottom of my heart for sparing her precious time to be part of this scientific get-together. Thank you, Vinita, once again. And now I express my sincere, heartfelt thanks goes to our principal, Dr. Jolie Andrews, Christ College, and manager, Father Jacob Nerinjambali, vice principals, Dr. K.Y. Shaju, Dr. Driven Father Joy Pinnacle Parambil, and Dr. C.A. Joshi, our head of the Department of Zoology, and Dean of Science, Dr. Tessie Paul, and Dr. Robinson Polministeri, our IQAC coordinator, and all other faculty members of the Department of Zoology who have been exceptionally helpful and supportive. Now, let me thank the research scholars of Smriti, especially Ajit V. Johnson, Ashwati, Adina, and Danusha, for their indomitable spirit that made this event reality. I would like to thank the technical team for their effective and efficient backup, making this virtual platform happen without any interruptions. A word of gratitude and appreciation to all of the participation for their cooperation and patience. Once again, my heartfelt thanks to all of you. Have a great day. Stay safe, stay healthy. Thank you.
Thank you so much.
Thank you, Vinita.
Thank you, Vinita. Thanks a lot.
Thank you.